Hello, welcome to How to Science 123. This is my first video. Today we're going to be talking about cellular respiration. So what is cellular respiration? So cellular respiration is the process that breaks down glucose, or C6H12O6, to make ATP or energy. It usually occurs in three main stages. We have glycolysis, pyruvate oxidation, which is a factor of the citric acid cycle, and then oxidative phosphorylation. So we'll be talking about two types of respiration. We have aerobic and anaerobic, but for this section, we're going to be just specifically talking about the one that requires oxygen, or the aerobic respiration. And this is the formula for cellular respiration. We have glucose and oxygen turning into carbon dioxide, H2O, and also 36 ATP. So before we really dig into cellular respiration as a whole and we dig into all the specific steps of it, I want to talk about redox reactions. Um, so what exactly is a redox reaction? So a redox reaction is the process of oxidation and reduction, right? If we combine the two, we get the term redox. So we have two parts to a redox reaction. We have something that's getting oxidized and we have something that's getting reduced. So in the terms of oxidation, we are losing electrons, and in this case, when we're usually talking about that, um, especially in biology, that refers to losing a hydrogen. And when we're reducing or we're gaining electrons, it means we're gaining a hydrogen. So how does gaining a hydrogen correlate to reducing, and how does losing a hydrogen correlate to oxidizing? So in the terms of reduction, right, if we're gaining electrons, we're becoming more negative. So if we're becoming more negative, then we're becoming smaller. And in the terms of oxidation, if we're losing electrons, we're becoming more positive, we're losing that hydrogen, so we're becoming a little more bigger. And a little cool term that I like to use in order to remember how all of these things work. Leo the line says Gur. So in this case, Leo represents losing electrons equals oxidation, and Gur represents gaining electrons, which equals reduction. So in the terms of cellular respiration, right, if we go back to our original equation here, right, this glucose is going to get oxidized, right? It's going to lose electrons, and the oxygen is going to get reduced. It's going to gain electrons. And this flow of electrons is what drives ATP production. It's going to be very, very important when we get to things like electron carriers and the next few slides. This is a pretty simplified diagram into how all of this all works. So in this case, we have this A atom or this A molecule, and it's going to oxidize. It's going to lose electrons, right? But these electrons, right, they need to go somewhere, right? The electrons just don't vanish into thin air. They don't just, go nowhere. So pretty much that's why if we're going to oxidize something, then we're always going to reduce something. That's why they're referred to as redox reactions, because oxidation and reduction go hand in hand. And same, if we're going to reduce something, then we're always going to oxidize something. So in this case, we'll just say atom, this A atom is going to oxidize. It's going to lose an electron. And in return, that electron is going to go to this B atom, and it's going to reduce that B atom, right? Because if we're gaining electrons, we're becoming more negative, we're becoming smaller. And if we're losing electrons, we're becoming more positive, we're becoming bigger, and that refers to oxidation. In the case of cellular respiration, it will have so much to do with hydrogens, because hydrogens, they're going to get, transferred in and out of. There's just going to be a bunch of redox reactions that we'll be going over in the next few slides. So to further drill down the concept of redox reactions and the terms oxidation reduction, we also need to understand what an oxidizing agent is and what a reducing agent is. So it's kind of confusing because you see an oxidizing agent is something that's getting reduced, it's gaining electrons, and then you see a reducing agent is something that's getting oxidized or losing electrons, right? Why would the reducing agent be getting oxidized, and why would the oxidizing agent get reduced? So I like to use this a good image on methane and or the oxidation of methane into carbon dioxide and water. So we see methane here, right? It's the reducing agent. And why is it the reducing agent? Because it has, all of these electrons, right? It has all these hydrogens. And it's going to get oxidized. It's going to lose that hydrogen and turn into carbon dioxide, right? It's going to oxidize methane. The loss of hydrogens is going to oxidize methane and turn it into carbon dioxide, right? But if we're going to oxidize something, then we're always going to need to reduce something. So those electrons, they're going to transfer over to this 2O2 and help create 2H2O, or obviously water, right? Methane, right? It's the reducing agent, right? Something needs to lose electrons in order for something to gain electrons. Oxygen, it's the oxidizing agent. As you can see, it has no hydrogens attached to it, right? It's going to want those electrons. So this methane is going to reduce itself, excuse me, it's considered the reducing agent because it can donate electrons and therefore become oxidized. And then oxygen is going to take in those electrons and become reduced. It's a little confusing, I understand. And this whole process of reducing and oxidizing agents is imperative when we get to things like electron carriers such as NAD+, FAD, and O2, and obviously reducing agents such as glucose and NADH, right? And a key thing that I like to emphasize when we're looking at these kinds of things is following the hydrogens and oxygens, right? If we look at our products and we see one part of the products has hydrogens, then we know that it got reduced, right? And if we look at the other part, right, if something is getting reduced and the other part of the products had to have gone oxidized, if we see something that has oxygens on it, then we know that this is going to be something that got oxidized, right? So just follow the hydrogens and oxygens on the sides of the products to determine what got oxidized and what got reduced, right? And on the reactant sides, we determine what's considered the reducing agent and the oxidizing agent. CH4 is always going to be referred to as the reducing agent because it has the hydrogens. And then 2O2, right? If this is the reducing agent, we can't have two reducing agents. 2O2 is going to be considered the oxidizing agent because it has no hydrogens, right? It's wanting electrons. And so we know what's considered reduced, we know what's considered oxidized, and we now know what's considered a reducing agent, and we now know what's considered an oxidizing agent. So we connect the two, right? The reducing agent's going to get oxidized, so the reducing agent, which is CH4, will get oxidized into CO2, and then the oxidizing agent, which we determined is 2O2, will become reduced into 2H2O. If that makes any added sense, hopefully it does. All right, let's get into the first step of cellular respiration, which is glycolysis. So some quick, important facts about glycolysis. It's located in the cytoplasm. People, when they think about cellular respiration, they assume that all of it happens within the mitochondria. That's not true. Glycolysis actually occurs outside of the mitochondria, and then the rest of the steps usually occur within the mitochondria. So the process of glycolysis is breaking down one glucose molecule into two pyruvate molecules. And it produces 2 ATP, it produces 2 NADH. There's no oxygen needed. This is an important fact. Glycolysis is anaerobic. It doesn't require any oxygens. And think of it as the entry point to respiration. So this is the first step in cellular respiration. In order for any other step to work, glycolysis needs to happen. So I like breaking down glycolysis into two phases. We have the energy investment phase, and then we have the energy payoff phase. All right, we can talk about the energy investment phase right now. So why is this phase referred to as the energy investment phase, right? Why is this phase referred to as the energy investment phase? If we just look at the two first words, energy investment, right? We're investing energy into a process. We're giving energy to have something else happen, and that something that happens occurs in the energy payoff. And the most common form of energy that occurs in our cellular processes is ATP, right? We are investing, in this case, ATP into these, the step 1 and step 3 to help us create this. We have a G3P molecule and we have a DHAP molecule. And these two molecules will go into the energy payoff phase to help create a pyruvate, right? So we are investing 2 ATP and in return we're getting 4 ATP, right? If we look in the energy payoff phase, the 2 ATP is going to turn into 2 ATP. So the net total, or the net ATP produced from glycolysis, is 2 ATP. And why do we want to track? Why do we want to track the ATPs? Well, if you go back to our first slide here, right, we have the cellular respiration formula. The most important aspect of cellular respiration is ATP, right? Cellular respiration has everything to do with the mitochondria, and we want to know how we get the energy for us to do anything, right? 36 ATP is a lot of ATP. So we know that we don't get a lot of our ATP from glycolysis, but we want to track those ATPs because they're going to be very important when we get to the later steps of cellular respiration. But let's go more in depth into glycolysis. So there's some key enzymes that I feel like I want to emphasize in this. Any enzyme that you see with the term kinase next to it is relative to phosphorylation. And what does that mean? Phosphorylation refers to the transfer of phosphates, right? This ATP molecule, right? This hexokinase enzyme is essentially what it's going to do is it's going to take this high energy bond phosphate on this ATP and it's going to transfer, right? We talked about the kinase enzymes, being used in the transfer of phosphates. It's going to transfer this high energy phosphate bond to this originally what it was a glucose molecule and turn it into glucose 6-phosphate. And we refer to this as substrate level phosphorylation, right? It's phosphorylation happening at the substrate level. So it's basically happening in each step of the process, right? We have phosphorylation occurring here in the energy payoff phase, and we have phosphorylation occurring, here in the energy investment phase. So anytime you see a kinase enzyme, you know that you're dealing with something phosphorylated. You're dealing with the transfer of phosphates. And this is very important, right? These kinase enzymes are very important because they help these phosphates. They basically drive a lot of the functions that occur in glycolysis, right? If we don't have the kinase enzyme, then this ATP, this high energy phosphate bond, won't be able to attach to this glucose molecule and create glucose 6-phosphate. So it is very important. And it's not just that. Phosphorylation doesn't, it's the transfer of phosphates. You could transfer 2 ADP, these two phosphates, to create 2 ATP. So it's vital. And remember how we were talking about the redox reactions, which were the transfer electrons or the movement of electrons, right? We refer to as kinase being the transfer of phosphates. Well, dehydrogenase is the enzyme that helps us transfer electrons to redox reactions, basically. So what do I mean by that? So let's look at this. We have 2 NAD plus 4 electrons plus 4 hydrogens, which will give us 2 NADH plus 2 H plus, right? We are going to be, what we're going to do here is we're going to basically, it's 2 NAD plus right here, right? If we're talking about redox reactions, it means we're reducing something and we're oxidizing something, right? In this case, this triose phosphate dehydrogenase is going to take hydrogen from here and transfer it to this NAD plus to give us 2 NADH. So it's going to reduce this 2 NAD plus and turn it into NADH, right? We're taking electrons, we're oxidizing this, we're going to oxidize this G3P molecule, right? We're taking electrons and in return, we're going to reduce NAD plus to turn it into NADH. So dehydrogenase is a very important enzyme because it basically helps us transfer hydrogens, right? We said in redox reactions, we're always going to follow the hydrogens and the oxygens. And dehydrogenase is a very important enzyme because it's going to take a hydrogen from this G3P molecule that we produced at the end of the energy investment phase, and we're going to give it to this NAD plus to turn it into NADH. And what is NAD plus? NAD plus is an electron carrier. It's an electron carrier. It's there to take the hydrogens. And this NADH will be very important when we get to the last phase in cellular respiration, oxidative phosphorylation. So in return, what do we get out of all this, right? By the way, each one of these steps is representative of one pyruvate molecule, right? Because we turn one glucose into two pyruvates. So at the end of this, one glucose molecule gets us two pyruvates and two H2Os. We get two ATP formed, right? Because we invested two and we gained four, so 4 minus 2 is 2. And 2 NADH plus 2 H plus, right? This 2 NAD plus the 4 electrons, it's going to take on the hydrogens, and this 2 NADH is going to take that electron to wherever. It's going to take it to the last phase or the last step in cellular respiration. As for this 2H plus, it's going to be important as well later on in terms of other things, which we'll get into later. But glycolysis is a very important phase because this is where everything begins. So pyruvate oxidation. Pyruvate oxidation is tentatively the most, I wouldn't really say, okay, it's not useless. It's very important, but it's the most like uncategorized step. It's usually a part of the citric acid cycle, but it's important to go over it specifically. But basically, right, we see the term oxidation here. If we're oxidizing something, we're going to be reducing something. So, right, at the end of glycolysis, we had one glucose, which we turned into two pyruvates. So now the pyruvates are going to enter the mitochondria. They're going to enter the mitochondria to get to the citric acid cycle. So this pyruvate is going to be converted to acetyl-CoA or acetyl-coenzyme A before entering the citric acid cycle. So we see this pyruvate molecule, right? It's going to use a transport protein. It's going to lose a carbon dioxide. It's going to lose a carbon dioxide, so it's going to, what do you think is going to happen? It's going to, right? It's going to oxidize itself. And in return, we're going to reduce something. We're going to reduce NAD+, right? We're oxidizing pyruvate. We're going to take this enzyme, the pyruvate dehydrogenase, which we talked about earlier. It's going to take a hydrogen and it's going to give it to this NAD+. It's going to reduce NAD+, and turn it into NADH. And if we're going to reduce something, then we're going to oxidize something at the exact same time too. So we're oxidizing pyruvate. Pyruvate's going to lose the carbon dioxide, and we're oxidizing pyruvate by taking away the hydrogens and giving them to NAD+ instead. And so after all of these processes happen, coenzyme A is going to come in, and now we have this new structure. We have C double bonded to O, CH3. Remember, the CO2 is gone, right? Because we oxidized the pyruvates of the carboxyl group, which released the first CO2 of cellular respiration, and that's what caused the reduction of NAD+ to NADH. And the combination of those two carbon fragments with coenzyme A allows us to form acetyl-CoA, right? We have this two carbon fragment, which we were talking about earlier, and then we have coenzyme A coming in and attaching to that. And this will be very important when we get into the citric acid cycle. So here's the citric acid cycle, right? So this is coming straight out of pyruvate oxidation. We have our acetyl-CoA. So our acetyl-CoA will go into the first step, and we're going to lose that coenzyme A. That coenzyme A is going to go away. but we have oxaloacetate, which was the last step, right? Remember, this is a cycle. So this is going to keep going around and around and around and around for each pyruvate. So 2 times, basically. So this acetyl-CoA will leave, and now we have this carbon structure. It's going to attach to this oxaloacetate. So now we have formed a 1, 2, 3, 4, 5, 6, a 6-ringed carbon structure, which is referred to as citrate. So we took these 2 carbons and we attached it to this 4-carbon structure to create citrate. So now there's going to be some changing of the shape. We're going to lose a water and we're going to gain a water. And now we've created isocitrate, right? Now watch what happens. We're going to lose a carbon dioxide. We're going to oxidize ourselves. And in return, we're going to reduce something. We're going to reduce NAD+. So imagine a dehydrogenase enzyme is here. It's going to take a hydrogen and it's going to give it to NAD+, reducing it to NADH. So now we go from 1, 2, 3, 4, 5, 6 carbons to 1, 2, 3, 4, 5 carbons. So 6, 5. Now we're going to lose another carbon dioxide. And if we're going to oxidize, then we're going to reduce. So another dehydrogenase enzyme is going to come in here. It's going to take a hydrogen from ketoglutarate and give it to NAD+, reducing it to NADH. And in the same step, we see this coenzyme A come in again. And this coenzyme A is going to attach to now what we have a 4-carbon structure, right? And we're going to keep it at 4 carbon. We're going to keep it as a 4-carbon structure all the way to step 8 because remember, oxaloacetate only had 4 carbons. So we're not going to lose any more CO2 after this. So this acetyl-CoA, this acetyl-CoA or the succinyl-CoA is going to leave. It's going to leave now, and this inorganic phosphate is going to help us produce ATP. So we produce 1 ATP per pyruvate, so per turn. So the citric acid cycle produces even less ATP than glycolysis. And at this point, how much ATP do you think we've produced? 4 ATP, right? Because we produce 1 ATP for each turn of the cycle, and if there's 2 pyruvates, we have 2 ATP total produced. So far we've only produced 4 ATP, and we need 36 for for cellular respiration to actually be beneficial for us. So we have not produced a sufficient amount of ATP, but we see the same process here, right? ATP is used, the CoA leaves, and so now we're going to reduce, right? We oxidize succinate. We're going to take a hydrogen from succinate and give it to FAD, which is another type of electron carrier. So we have 2 types. You have NAD+, and we have FAD, and it's going to give it to FADH2. H2O is going to come in. It's going to change the shape of fumarate and turn it into malate. And then we see a similar situation here. We're going to take an electron and we're going to give it to NAD+ to create NADH to give us oxaloacetate. And now the second pyruvate is going to come in and repeat the cycle all over again. So to summarize, a citric acid cycle, per turn, we produce 3 NADH, 1 FADH2, 1 ATP. And then we also lose 2 CO2 in the process. So in total, for 2 pyruvates, we only produce 2 ATP, we produce 6 NADH, we produce 2 FADH2, and we lose 4 CO2 for those 2 pyruvates, right? Because the citric acid cycle is going to turn twice. So where's the rest of the ATP, right? Where is the rest of the ATP coming from? Where's that? And this is where we get into the most important step. Oxidative phosphorylation. Oxidative phosphorylation is the most important step because this is where all our ATPs come from, right? There's two parts to oxidative phosphorylation. We have the electron transport chain and then we have ATP synthase. So essentially what we're going to do here in the electron transport chain, it's in the name, all the electrons that we had, right? We reduced NAD plus, we turned it to NADH. All of these electron carriers, they're going to take all their electrons to this one step in cellular respiration referred to as the electron transport chain. And it's going to go through a process of just oxidizing. It's going to go through a bunch of redox reactions basically, right? Because essentially we're going to lose an electron in terms of oxidizing NAD plus, and we're going to keep doing that down and down and down the electron transport chain till eventually we have an electron acceptor, which is oxygen 1/2 O2, right? It's going to want those two electrons to turn into a full oxygen and give us H2O. So oxygen, the end of all of this chain of just redox reactions of just, transferring electrons over and over again. Eventually, we will have our final electron acceptor. We have a 2H plus plus a 1/2 O2. It's going to want those two electrons to give us H2O. Remember, we talked about this H plus here, right? This H plus, you know, just floated around the area, just waiting to do something. That's where this H plus comes into play. It comes into an even bigger play in a little bit, but it's very important here as well because it helps us produce one of our first products, or our second product actually. We produced a lot of CO2 earlier, but now we produce our second product, which is H2O. But this is, obviously we looked at this here, electron transport chain. Where is the ATP being produced? We still haven't produced any ATP. This whole process here didn't give us a single ounce of ATP. It gave us water, but it didn't give us any ATP. So where does the rest come into play? That occurs here in ATP synthase. So essentially, the energy released as electrons are passed down the electron transport chain is used to pump H plus from the mitochondrial matrix to the intermembrane space. So if we look back here, as these are all being, as the electrons are being passed, NADH is being oxidized into NAD plus. The electrons, or excuse me, these H plus protons, which are on the inside, they're going to use that energy of this redox reaction to pump themselves outside of the cell. So what does that create? Well, we talked about the electrochemical gradient last time, or if you know about the electrochemical gradient, excuse me, we have essentially created a more positive gradient, right? We have a surplus of H plus ions outside of the cell than we do inside of the cell. So there's a more positive gradient and there's a more negative gradient, and opposites attract. So essentially, these H plus ions, they're going to be attracted to this negative inner, they're going to be attracted to the negative charge, which is found inside the mitochondrial matrix, and they're going to go through this gigantic enzyme referred to as ATP synthase, and essentially it's going to turn a turbine. It's a very, very cool structure, basically. It's going to turn a turbine, and it's basically going to catalyze a reaction that creates ATP, right? The H plus moves into the binding sites on the rotor of ATP synthase, which causes it to spin in a way that catalyzes phosphorylation of ADP to ATP. And this is an example of chemiosmosis, the use of energy in an H plus gradient to drive cellular work, right? There is a positive surplus of H plus outside of the cell, and there's a negative gradient of H plus inside of the cell. There's less H plus inside than there is outside, so it's more positive on the outside. And that's what's going to cause this H plus to say, I want to come in, I want to go into that negative gradient. And it's going to go through this ATP synthase, which is going to turn these rotors and it's a very cool structure. It's going to turn these rotors to essentially power or catalyze the phosphorylation of ADP and that inorganic phosphate into ATP. And this is pretty much where we produce all our ATP, right? How much did we produce? We produced 2 ATP in glycolysis. We produced 2 ATP in the citric acid cycle. So we only produced 4 ATP prior to oxidative phosphorylation. But essentially what this did, what the electron transport chain did, is it helped create this positive gradient of H plus outside of the cell in order to have oxygen be the final electron acceptor. But it created this positive gradient of H plus outside of the cell, which we see here, and this more negative gradient inside the cell to then therefore cause the H plus to drive back into the cell, right? Because they want to go to a more negative gradient, right? Opposites attract. Go through the ATP synthase, turn around these rotors and catalyze a reaction that turns ADP into ATP. And this is producing a large amount of ATP. 34 ATP, 32 ATP, a large amount of ATP. So the rest of our ATP, right, we finally have our final products. We had our CO2, then we had our H2O, and now we have our approximately 36 ATP that are produced from just this one step of the oxidative phosphorylation. So there's two big steps in oxidative phosphorylation. We have the electron transport chain, and then we have ATP synthase. This is an image that sort of summarizes everything here. I think this is the best image. But basically, right, the NADH, it's going to reduce itself, it's going to oxidize itself back in NAD+, and something needs to take on those electrons, and this protein complex of electron carriers is going to take those electrons, and it's going to pass it here, right? So this complex will get oxidized, and this will get reduced, and this will get oxidized, and this will get reduced, and this will get oxidized, and eventually this oxygen will get reduced, right? It will become the electron acceptor, the final electron acceptor, turning it into H2O. And remember, we had the FADH2, which we looked at earlier. It's the same process here. The FADH2 will oxidize itself into FAD, right? Those electrons are going to need to go somewhere else. So this Q complex is going to take those electrons, and then it's going to oxidize itself, and this 3 complex is going to reduce itself, and then the 3 complex is going to oxidize itself and give its electrons, or the electrons from FADH2, to the cytochrome C complex, and then it's going to once again oxidize itself and give its electrons to this 4 complex. And then finally, oxygen is going to take on those electrons because it's the final electron acceptor. It's going to reduce itself and turn into H2O because we have, you know, we have some H plus inside. So H plus inside the mitochondrial matrix. And through each of these steps, the H plus inside of the cell, this is going to produce some energy that allows this H plus to just drive itself outside of the cell. So the electron transport chain, it's a whole separate process. It doesn't produce ATP, but it helps drive the process in oxidative phosphorylation that gives us our ATP, right? The electron transport chain produces some variable amount of energy that, right, we have some H plus, we have H plus inside the mitochondrial matrix, but it's going to cause the mitochondrial matrix to lose some of that H plus. The H plus is going to leave the mitochondrial matrix and go into the inner membrane space. And eventually, right, there's going to be a surplus of H plus outside of the cell and less H plus inside of the cell. So we have a more positive gradient outside and a more negative gradient outside. And it's going to cause that H plus to go through the ATP synthase, turn some rotors, catalyze a reaction that produces ATP. And this is referred to as chemiosmosis. So very, very long and lengthy process, as you guys can tell. But all of this essentially gives us our 36 ATP. But as the slide says, there are some exceptions. Fermentation. So obviously, we talked about aerobic respiration, and that's when oxygen is the final electron acceptor. But in some cases, our body, right? If we're really, really tired, and let's say we ran 400 meters, a 400-meter sprint, eventually our body can't take in any more oxygen, right? It can't, you know, we can't, we can't keep inhaling more oxygen. And so eventually, right, if there's no oxygen, then what's going to be the final electron acceptor in the electron transport chain? If there's no oxygen, our body needs to figure something else out. And this is where anaerobic respiration comes into play. So, for example, some organisms, they use a sulfate ion as a final electron acceptor. And in this case, hydrogen sulfide is made as a byproduct instead of H2O, right? So instead of the oxygen coming in and taking in that final electron, it's a sulfate, which gives us H2S. But in terms of humans, glycolysis oxidizes glucose to pyruvate without the involvement of O2 in our electron transport chain. Remember, we said glycolysis does not require an oxygen. It has no need for that. It's an anaerobic respirator. So in this case, NAD plus is referred to as the oxidizing agent. It's the one that's going to oxidize itself, and glycolysis produces 2 ATP by substrate level phosphorylation, regardless of whether O2 is present or not. So if you go to, for example, an example of fermentation is alcohol fermentation. If you like beer, this is how beer is made. So we have glycolysis, which produces our 2 ATP, like we said, and it gives us our 2 pyruvates, right? 2 pyruvates are going to lose the carbon dioxide. The NADH is going to essentially, uh, it's going to take on those electrons, and then it's going to reduce itself or give those electrons away, and essentially NAD plus is created. And that gives us NAD plus regeneration, which gives us ethanol. And it just repeats that cycle all over again. Lactic acid fermentation is also, we say lactic acid buildup when we're really tired. It's because we're going through anaerobic respiration. We have a lot of lactic acid buildup in those parts. So the important understanding between alcohol and lactic acid is that there's no release of CO2 in lactic acid fermentation. Like we said, these 2 pyruvates, they're going to lose 2 CO2 and then turn into 2 acetaldehyde. But in this case, there's no CO2 lost. So glucose is going to go through glycolysis. The NADH is going to, it's going to reduce itself. It's going to lose those electrons. NAD plus is going to get oxidized instead, and we in turn produce 2-lactate. So, which will then give us more NAD plus regeneration. So this is the process of lactic acid fermentation, and the key differences: fermentation and anaerobic and aerobic respiration have some similarities. They all use glycolysis, producing a net ATP of 2 to oxidize glucose and to harvest the chemical energy of food. In all three, NAD+ is the oxidizing agent that accepts electrons during glycolysis. One major difference is the mechanisms used to oxidize NADH to NAD+. In fermentation, an organic molecule, pyruvate or acetaldehyde, acts as a final electron acceptor. And in cellular respiration, electrons are transferred to the electron transport chain.